We hope you guys had a good lunch and you enjoyed it. First, we would like to remind you that tonight, following the symposium, we'll be having a social event at 6 p.m. at Cafe Excel, and that's in Bryan, Texas, on Texas and University. So there will be pizza and appetizers provided free of charge, and there will also be a cash bar available for you guys to come socialize and get to interact a little bit more. So we will be at this event. We will be having the presentation of the winners of the poster session, as well as acknowledging several of the traveling oral speakers, as well as the Texas A&M speakers. So it's a great opportunity for everyone to come during the evening and meet and socialize with keynote speakers and the rest of the general audience in the crowd. So we hope that you come. We invite you to bring your significant others along with as well and come have a great night with us. Okay, so now we will hear from our third keynote speaker of the day, Dr. Keerti Rathore. Dr. Rathore is professor in the Department of Soil and Crop Sciences at the Texas A&M University. I don't have it. Sorry. So he is a professor in the Department of Soil and Crop Sciences at Texas A&M University. His research focuses on genetic improvements of crops through the development of protocols for efficient delivery of genes, optimal expression of transgenes, rapid recovery of transgenic cotton, rice, and sorghum. His projects include regeneration from cell and tissue cultures, use of new reporters and selectable marker genes to understand and improve transformation processes, promoter analysis, enhancement of disease resistance in plants, conferring drought tolerance in crop plants, conferring insect resistance in crop plants, improving nutritional quality of seeds, and production of recombinant antibodies and vaccines in plants. His lab has recently applied for deregulation of a transgenic cottonseed with localized suppression of gossypol, making his lab one of the handful of public programs that has filed for release of a deregulated transgenic crop in the public sector. Additionally, his group is working on root rot nematode resistance and development of CRISPR-Cas protocols for cotton. Dr. Rathore holds a BS in zoology, botany, chemistry, and a master's in plant science and a PhD in plant physiology. The title of his talk is From Conception to Towards Deregulation of Ultra-Low Gossypol Cottonseed, a 20-year odyssey. Please help in joining Dr. Rathore.
Thank you for the introduction, Steve, and thank you, the organizing committee, for selecting this particular talk, which is my favorite talk for the last 20 years or so, and it's been a long journey, but at least as far as taking an idea to the product stage, I think we're pretty close, hopefully. And also, thank you for sticking around after lunch. Usually, once people have had their lunch, they leave. So thank you for sticking around. Now, this is a subject that many people who are not in the cotton area would know little about. So I'll have quite a few introductory slides, so to give you some background as to what the problem is and why I got involved in this and why we spend so much time on this thing. So if you're like me 20 years ago or 22 years ago, you wouldn't think that cotton plant produces almost 1.6 times more seed than it produces fiber in terms of weight. So overall, worldwide, we are producing almost 24 million metric tons of fiber. Along with that fiber, we produce about 47 million metric tons of seed. Now, cotton seed has about 22-23% protein. So that means that in terms of cotton seed production, we are producing about 10 to 11 million metric tons of protein every year. And it's a good quality protein. Now, a human being requires about 50 grams of protein per day to survive. Now, if you could utilize that protein directly for human nutrition, you could meet the basic protein requirements of anywhere between 550 to 600 million people. And that's a lot of protein that is right now not being utilized in a very efficient way. And the main reason for that is that cottonseed contains this compound, terpenoid gossypol, and gossypol is toxic to monogastric animals, including human beings. And so this is this was one of the impetus for us to get started on this project. You may be looking at this little mountain. It's cottonseed just piled up right outside Lubbock. And just to get some perspective how big that is, here is a pickup truck. You can see that. So there's a lot of cottonseed produced all around the world, and it'll be nice if we could utilize this cottonseed as a source of protein. So as I mentioned, the protein content of cottonseed is about 22-23%, and it's relatively good quality protein. And as I said, monogastric animals or human beings cannot use it because of the presence of gossypol. Now, the gossypol is produced and stored in these dark color glands that you can see in the seed. This is a slice of a seed showing these glands as the structure of gossypol here. Now, as I said, gossypol is toxic to monogastric animals such as pigs, chicken, fish, and so on. The only animals that can handle gossypol, and that to a certain level, are the cows, the cattle. And we do utilize cottonseed oil. So cottonseed is crushed to get oil. Oil is refined, and through some chemical and physical processing, gossypol is removed. And after that, it's pretty healthy, and we all eat cottonseed oil. This slide shows the countries, some of the countries that produce cotton. So here are the 4 major producers and their production levels as well as the export levels. Here are some of the African countries, West African countries that also produce cotton. And cotton is a big crop there. These numbers in the red really show the export income, the fact that cotton export is number 2 earner of the export income in this country, Burkina Faso, number 1 in Mali, and so on. And some of these other countries in Africa that also produce cotton. But look at the fertility rate. These are the countries where the population is rising very fast. There is already protein malnutrition, and they're going to face much more severe problem as time goes. Now, a lot of these countries that produce cotton basically survive on some of these cereals. That's the staple food for them. And the level of protein that these cereals have is much lower than what you normally want in a diet. A lot of countries, people just survive on cassava or yam that have extremely low levels of protein. By the way, the project that I'm going to talk to you about today, Dr. Borlaug, he was in our department. He was one of the biggest supporter of my project. He knew the value of this project. U.S. farmers are also very interested in this particular product, or gossypol-free cottonseed. And in fact, a survey was done in 2015, and they put improved cottonseed value as number 4 item on their list of what they wanted. And obviously, if the cottonseed becomes a source of food or even as a source of feed for some of the monogastric animals, its value will go up, and cotton farmers will get more value for their money without doing anything additional that they do now. So as I said, we do utilize cottonseed oil in our diet directly, human nutrition. Also, so we're not throwing away the cottonseed. We're still using it before or after oil extraction as a feed for cattle now, and we obviously cannot use it for some of these monogastric animals. So what's wrong with that? Somebody will say, okay, we're still using it indirectly for human nutrition. The fact is that cows are not the most efficient animals. So a milk cow produce, I mean, it has to, you have to give it 3.66 kilograms of crude protein to get 0.83 kilograms of milk protein. And the protein conversion ratio for beef cattle is 20, meaning that 20 pounds of crude protein to get 1 pound of beef protein. So very inefficient animals. Compare that to some of these other animals. So here is pig, chicken, 5 times more efficient. Eggs are the most efficient way to convert crude protein into animal protein. And some of the fish that we eat more and more, the aquaculture grown fish like salmon, tilapia, are also much more efficient than the cows. So if you had a limited amount of protein, it's better to give it to some of these animals than to just simply feed it to the cows. So the problem has been known for a long time, and people have been trying to address it for a long time. And in 1954, McMichael discovered a glandless mutant of cotton. Now, as I said, gossypol is present within these dark colored glands that you see all over the cotton plant. And since the plant didn't have any glands, it did not have any gossypol. And there was a great deal of excitement during that time, and a lot of national and international projects were launched to incorporate this trait into commercial varieties of cotton. And at the same time, there were also animal nutrition studies were done, and they found that the cottonseed, which was gossypol free, was good enough to feed to the chicken, pigs, fish, and so on. In fact, it was considered good enough for human consumption. In fact, Texas A&M tried to to introduce these roasted and salted cottonseed kernels. They called it tamu nuts, and people actually like the taste of those tamu nuts. Now, so this is the situation. Is so most of the cotton we grow today, wild type cotton, is glanded. There are glands present in the leaf and other parts of the plant. And here is the seed. The seed gossypol level is about 10,000 parts per million. Here is a mutant glandless cotton, so no glands in the leaf or any other part, certainly no glands in the seed, and very low or negligible levels of gossypol. Now, gossypol that is present, and this is a picture showing the presence of glands in the leaf, in the sepals. It's also present in the petals, flower petals, ball, here, bracts tissue, stem. Gossypol present within these glands protects the plant from insects, and there is some reason to believe that it also plays a role in defending the plant against certain fungal diseases. Okay, so it's necessary for the whole plant. So once the farmers try to grow this glandless cotton in their field, their glandless cotton was attacked much more heavily by these traditional insect pests of cotton. There are insect pests that normally do not go anywhere near cotton. They were attacking the glandless cotton. So for that reason, there is no large-scale production of glandless cotton anywhere in the world right now, despite the fact that it proved highly effective in terms of as a feed for monogastric animals, as well as a source of nutrition for human beings. So what is the desired trait in cotton? So you want cottonseed to be low in gossypol. Now FDA considers cottonseed products safe enough even for human consumption if the levels of gossypol are below 450 parts per million. And but you obviously want levels to be low enough or lower than what FDA considers safe. At the same time, you want the normal levels and complement of these gossypol and other terpenoids in rest of the plant. So that was our goal when we started, and I started this project in '96. And we had to develop all the tools and technologies to conduct this project. So first, we obviously need an efficient transformation method. We need a target gene that we can silence so we can suppress the gossypol. We obviously need to do it in a seed-specific manner in order to make sure that the rest of the plant doesn't suffer from insect attack. And we needed a gene silencing mechanism. So we started this in 96, about 97, 98, we established this transformation protocol for cotton. And this was agrobacterium based method. Just to give you some idea how difficult that is, starting with the transformation of the cotton discs or stem or hypocotyl tissue to get to this stage when you get a transgenic plant in the soil takes about 10 to 12 months. So that's the time frame. But we had a pretty good system working in the lab. We needed to have a good seed-specific promoter, so we went after the seed storage proteins. And globulins are major seed storage proteins in cottonseed, and we figured that the promoter that drives the gene for this particular seed storage protein should be seed-specific, but we had to make sure that that is the case. So we isolated the sequence of the promoter and hooked up with the reporter gene GUS-A and transformed those constructs into tobacco, Arabidopsis, and cotton, and examine the activity of the promoter. So we find that in the developing cotton embryos, the promoter becomes active at around day 16 post-anthesis. And this is the higher magnification image of the same embryo. As you can see, this blue coloration here, that means that the promoter is just becoming active. And as the embryo grows, the promoter activity becomes stronger and spreads throughout the embryo. By day 25, the promoter is active throughout the embryo. And this is just about the day when the gossypol production begins in the embryo. Okay. We also made sure that the promoter is tissue specific. It's not active in any of these other tissues. The only place it's active is in the cotton seed. So, and this is the gene we are targeting. I'll tell you a little more about that in a second. But there is a good overlap between the promoter activity and the activity of this particular target gene. So when I started this work, the only gene silencing technology that was available or was known was the antisense technology. So we used this technology and made several constructs. Okay, this slide is an important slide. So what I want to say here is that this is the gene we targeted, delta-cadenine synthase gene. Delta-cadenine synthase is an important enzyme that's involved in the conversion of farnesyl diphosphate to delta-cadenine. And some of these biochemical steps were known at the time, and this gene was the first one that was isolated. So we isolated that gene from Gossypium hirsutum. The other thing I want to point out here is that these two parts of the plant, cotton plant, like the flower petals and the seed, the predominant terpenoid in the glands of these two parts is gossypol. The green parts of the cotton plant, like the leaf, stem, ball, rind, and sepals, and so on, the glands in those parts contain not just gossypol, but also hemigossypolone and heliocytes, and they're all derived from the same basic biosynthetic pathway. Some of these compounds have much higher insecticidal activity than gossypol. So we made some antisense constructs, and this was our several lines we derived. And these are the gossypol values in all these different lines. So these are T1 pooled T1 seeds. And we do find that some of the lines towards your right side, they are low in gossypol. So this is in T1 seeds. And we thought, okay, that's great. Let's look at what happens to the T2 seeds. And when we checked the T2 seeds, we got a big shock here. So the bars here in the red color, they are the values for the T1 seed. The bars in yellow or orange color here are T2 seeds. So what that told us is that the trait is not stable. We got some reduction in gossypol levels in T1 seeds, but not in the T2 seeds, except for one particular line here that showed us low levels and looked pretty hopeful. So we carried on up to about T4 generation with that particular line. And we did see some reduction in the level of gossypol in the seed. The levels in the leaf were similar, whether it's wild type or the transgenic cotton plant. So with the antisense technology, we were able to reduce gossypol in the seed to about 1700 ppm. So we came down from about 10,000 parts per million to about 1700 ppm. So we're nowhere close to what FDA considers safe, which is 450 ppm. So that was a big setback and almost 4 years of work. It couldn't even publish something like that. So we thought, okay, we will try. By that time, there were a few years after that, RNAi technology came along, and we thought, okay, can we utilize RNAi technology to do the same thing, which is supposed to be more powerful silencing technology. Now, before we even started our work on RNAi, there were several papers out there that showed that RNAi can spread from its point of origin to other parts of the plant. And they showed that in different ways, and probably no need to go into detail, but there were several papers that showed that if an RNAi occurs at in certain part of the plant, it can spread from that part to other parts of the plant. Now, if that was to happen in our case, where we are trying to suppress gossypol in the seed, the RNAi would have spread to other parts of the plant, in the leaf and other parts, and it would have silenced the gene and no gossypol. So we would be in the same situation as the as the glanless cotton. And in fact, some of the pioneers of this RNAi work, for example, Waterhouse. I think I lost the battery here. Okay. They wrote some reviews and talked about the fact that tissue and cell-specific RNAi may not be possible because of the spreading of silencing. Now, if we believed these papers, I probably wouldn't be standing here talking to you today. We went ahead and did the experiments anyway. And so again, the slide thing works, but this pointer thing is not working. Anyway, so basically we used the same promoter and we targeted the same gene, delta-cadmium synthase gene, and the only thing we did different was to use RNAi instead of antisense. Okay, next slide. Someone can. Okay, there you go. So we made we used about 600 base pairs of the trigger of the sequence as the trigger sequence. We made some hairpin RNAi construct. Next slide. Someone can. Yep. So we made the hairpin RNAi construct and again used agrobacterium. Okay, thanks. And use the agrobacterium to transform cotton. These are some of our, this is the result from our T1 seeds. So there are several lines here. Okay, the pointer thing still doesn't work. Okay, just the slides. Okay, all right. Yeah, so line number 232 and line number 81, these lines look pretty promising, and the levels of the seed gossipol in the pooled seed gossipol is shown. Yeah, sure. Okay, so we thought we'll go ahead and test these. So these are, remember, these are pooled T1 seeds. They would be segregating for the transgene. Okay. Oops, that'll do it. So we carried out southern blot analysis on these three lines, and we see that the transgene is certainly integrated into the genome. And this was our first result, which looked very promising. So here are individual 10 seeds from a wild type or non-transgenic plants, and these are the gossipol values in individual seeds. These are the two lines, line number 2 and 32. The seeds that had the gene, which is shown by this PCR result, had very low levels of gossipol. The seeds that did not have the transgene have normal levels of gossipol. So remember, these are T1 seeds that are going to be segregating for the transgene. So that was very promising result. And if you look at the HPLC chromatogram, normally the cotton seed that will show a big gossipol peak like that. And here are some of the seeds that are transgenic or expressing the RNAi construct. Can barely see the gossipol peak. Okay. And we obviously checked the levels of transcripts, and we showed that the gene that we were targeting was indeed silenced in these two transgenic RNAi lines. We confirmed that again with northern blot analysis, and they showed the same result. We've done the enzyme analysis for this delta cadmium synthase enzyme, and the enzyme levels are extremely low in the RNAi lines. So these are all, these results are from the embryos, developing embryos. So far so good. What happens to the leaf and other parts of the plant? And so we chose the same two lines, line number 2 and 32. Again, we chose 10 plants from, these are again T1 plants, so 10 plants from each, in each case. And these are wild type plants. Now remember the slide that I showed you earlier, that the green parts of the plant contain not just gossipol, but also hemigossipolone and heliocytes. So these are the values for gossipol, hemigossipol, and heliocytes. And as you can see that we don't see any reduction in the level of these terpenoids in the leaf. Here are some other parts that insects normally like to chew on, and the terminal bud, the floral bud, bracts tissue, petals, ball, and the root. Again, the red bar is wild type or non-transgenic cotton, and the two green bars are the two RNAi lines. And we see that the levels of these terpenoids are very similar, whether it's transgenic plants or the RNAi plant. So this these results show that the trait is totally tissue specific, that the reduction of gossipol we are finding is only in the seed and no other part of the plant. So we went to T2 generation to make sure that the trait was stable. And so this again, line number 2 and 32, these are T2 seeds from T1 homozygous plant. And all the 50 seeds that we tested have very low levels of gossipol. So this result showed us that the trait is stable and is transmitted to the progeny. So unlike the antisense work that I showed you earlier, which was not stable, most of the lines reverted back to normal levels of gossipol. This looked like it is promising. This was another third line that we had tested. Again, these are individual values of gossipol in these seeds. And if you average those out, it's about 190 parts per million gossipol in those seeds. and the wild type seeds have close to about 10,000 parts per million gossypol. So what happens if you take these lines to the field? So all this work that I had shown you earlier was done in the greenhouse stage. Things are different once you go to the field. We wanted to make sure that the trait was stable under field conditions. And so we have three different lines that that we had selected and tested those in field conditions in 2009, 2010, and 2011. And again, as you can see, compared to the non-transgenic, the levels are low. Seed gossypol levels are low. So the trait is stable under field conditions. So that's where we are. So we are now calling this trait ultra-low gossypol cottonseed. We were able to reduce the levels from somewhere around 10,000 parts per million to about 250 parts per million. So if you go from generation to generation, it's not going to be exactly the same number. It may be 200 sometimes 275, but overall we have about levels about 250 parts per million, which is way below what FDA considers safe, and also WHO, FAO. Their numbers are much higher. They're about 600 parts per million. Being safe for human consumption. But the main important thing is that the foliage and floral parts of the plant contain normal levels of gossypol and related terpenoids. So their defensive functions should not be compromised. So starting in 96 to about 2006, when we had our first publication on that, this was the situation. So this is again just to kind of reiterate what I said. This is commercial cotton, 10,000 parts per million of gossypol in the seed. Here is the leaf, and you can see the glands clearly. This is the glandless cotton, very little or no gossypol in the seed or any other part of the plant, and no visible glands. And this is our engineered cotton, ultra-low gossypol cotton seed. Values in the seed are about 250 parts per million, but the leaf and other parts of the plant have normal glands and normal levels of gossypol and terpenoids. So it looked very promising and looked like the trait is stable and it is inherited generation after generation. So in 2006, we started taking some steps towards deregulation of this trait. And we generated many more lines. We certainly cannot match what all you guys in DuPont Dow do, who create something like 1,000 lines or 2,000 lines for any given trait. But we were able to generate at least 300 or so lines. And from those, we checked them for number of copies. We did a lot of southerns and insert localization, silencing, made sure that the gene was silenced and the levels of gossypol were low and the trait was stable in field conditions and so on. Carried out a lot of biochemical analysis. We did inheritance analysis. Over 2014, 2013, 14, and 15, we carried out multi-location field trials in 3 different states. There were 8 different trials, and we looked at the agronomic performance of a couple of lines, and also we did the seed composition analysis on those. Obviously did the fiber quality analysis, and all these analyses showed that apart from the low level of gossypol, everything is very similar. And in September, October of 2017, we filed a petition to USDA APHIS and also submitted a dossier to FDA. Okay, so that's where we are, and maybe I shouldn't, you can all check the timeline, already gone through that. Okay, so here is where you can use this cottonseed. You can use it to feed these animals, which are normally very susceptible to toxin, or you could even use it directly for human nutrition. And aquaculture, which where I think that it could really meet the demand for aquaculture feed, because it's one of the fastest growing industries. It's growing at a rate of 8% per year. And there is already shortage of feed for the aquaculture species like salmon and tilapia and shrimp and so on. Skip that slide. Probably, yeah, this is just a very quick slide to show chicken meat production in India. India used to be a largely vegetarian country, and look at the level of the chicken production, almost nothing until about 81, and now it's skyrocketing. And the same sort of thing is happening in a lot of other poor countries where the middle class is rising and there is much more demand for meat and so on. Okay, yeah, just the last or second last slide here. So now every time you do something, the question comes up, can you use CRISPR to do the same thing? Well, CRISPR, okay, if you were to target delta cadmium synthase gene with CRISPR, uh, there is no way to do that. We find that in the embryo there are at least 10 genes that are expressing delta cadmium synthase gene. It's a member of a large gene family. Okay, so that part won't happen. Even if you can find a single copy gene in cotton that you can target to do the same job, which by the way we do have a gene that we think will be very good for doing the same thing. But you can target it, but you cannot make it seed specific. If you knock it out once, it's gone forever. Okay. Now, there is a possibility that one can use CRISPR interference to do this work, but again, your plant is still going to be considered transgenic, so you really don't gain anything by using CRISPR interference. So here are the countries where there is rampant malnutrition, all the countries that are shown in color here. Here are the cotton producing countries that suffer from malnutrition. So these countries can benefit a great deal from this particular product if they adopt it. And here are the people who actually did the work. So there are people in my lab, most of them are left, but the ones in yellow that are shown here, they're working on this project. We had a good collaboration with Dr. Wayne Smith and Steve Haig that are in our department. And one of the big thanks is to USDA ARS people, Bob Stepanovic and Lorraine and Al Bell. They were extremely helpful to us in terms of analyzing gossypol in the seed and other parts of the plant. Couldn't have done this work without them. And since 2006, we had a big support from Cotton Inc. to carry out this project. And there are some of the other collaborators. In fact, Sam Reddy is in Dow right now, and he was involved in the early part of the promoter isolation and so on. So, and these are people who are contributed funding. And with that, I will stop and be happy to answer any questions.
Okay, thank you, Dr. Rathore, for sharing your story with us. So we have a few minutes for questions. Remember that if you have a question, please raise your hand and we'll bring you a microphone. So I guess we can start with Julian.
Do you know if this gossipol is only de novo synthesized in seeds, or can it get transported?
The seeds are, the gossipol that's present in the seed is produced in the seed. But it does other parts of the plant. Each and every cell is capable of producing gossipol. A lot of times, gossipol and these other terpenoids it comes in response to attack by insects and so on. Insects or some disease, fungus or some other disease. But the gossypol that's produced in the seed is coming from the seed, is produced in the seed itself.
Flora and the foliage path. Right?
No, we are not lowering the levels of gossypol in foliar tissue and the leaf or anywhere. That was the slide that I did show.
Okay, so my understanding, you can reduce the gossypol level in the condensate, but the leaf still has some high level, lower level.
It's the same level as the parental plant in the leaf.
So I thought you can keep the foliage or the floral path at a normal level to protect the plants against the insects. So if you can separate them, so my question is they controlled by the different genes or the same gene control?
Sorry, I don't fully understand your question. So the seed gossypol levels are the only ones that we have been able to reduce. All the other parts of the plant have the same levels of gossypol as the parental plant. So we have not either lowered or raised the levels of gossypol and terpenoids in other parts of the plant. So it's highly tissue-specific trait.
Excellent presentation. The question I have is if the human being consumes on a constant basis this source of protein, what are the chances of getting toxic if they consume and if they exceed the limit of the FDA regulation?
Yeah. Again, I did point out that you can use this for human nutrition. Personally, I think that is a much better source of. It's better to use this as a feed for animals, aquaculture species, and poultry, for example. As I said, there were a lot of studies were done in 60s, 70s, and that's where FDA determined this level of 450 parts per million to be safe even for human consumption. So nobody is going to eat this day in, day out, right? Or whenever people use it, it'll be used as a supplement to whatever else they're eating.
Right. Dr. Rathore, we have a question from the webinar from Colden Joy. Right. What do you believe is the biggest barrier to commercialization? Why has it not been picked up by the industry?
Maybe these guys can answer that. No, I'm joking. Well, I would be the happiest person. Or 10 years ago, we knocked at the doors of Dow, Bayer Crop Science, Monsanto, and well, obviously we were not successful in persuading them that this is a worthwhile technology. I guess this is my, and maybe one of you guys can answer later on. I guess they did not see how they can recover the investment by investing in this technology, and it is a big investment.
So I guess this is a follow-up to what Dr. Patel was asking. So how is this gossypol metabolized in animals or humans? Like, is it broken down, or if you keep consuming it, does it accumulate like on the skin or any other body parts?
No, it'll get metabolized. It'll get broken down. I mean, again, there is a limit. Everything is. There are a lot of toxins we consume, basically, but I mean, your body does break it down, and I think all these things have probably been looked at by FDA earlier on, many decades ago. By the way, I was talking about aquaculture. We have done 2 aquaculture feeding studies. We have fed this cottonseed to shrimp, and they grew quite happily and no problem. In fact, and this was done in Corpus Christi with some of our collaborators. We brought some shrimp back here. We analyzed the shrimp again with the help of our collaborators, Bob Stepanovic in his lab. And that shrimp that was fed on the ultra-low gossypol cottonseed, we saw no traces of gossypol in that shrimp. So it's safe enough for people to eat, even without removing the vein, which normally people don't remove that. And so even then, we could not detect any gossypol. So what I'm trying to say is that people do not have to eat cottonseed. The other study that we've done is on southern flounder. So that study also found that it's a very good source of protein for southern flounder.
I have another question from the webinar from Benjamin Merritt. Have you looked at the mechanism that is possibly causing the RNAi from translocating to other parts in the plant? In your case, as opposed to other cases where it did.
Yes, yeah, no, we have spent a great deal of effort trying to figure out, well, whether that was the case or not. And some of the studies that I did not show here, they were done using this grafting technique. So they had a silence plant, and they took a stalk of that, an unsilenced scion or something. They did a reciprocal grafting and showed that the silencing moves up or down. We've done exactly the same thing in cotton. We do not see any sign of spreading of silencing. The other way to look at this, or indirect way of making certain that that does not happen, is that, and I showed you one of the slides where we were looking at the segregating generation T1 seeds, and the seeds that did not have the gene were showing normal levels of gossypol. So if the RNAi was to spread, it would have spread from those silent seeds to these null segregants, and it did not. And we've done that in many different ways. So we don't know why cotton is different, but the early studies that showed this kind of spreading were done in tobacco and in Arabidopsis. So we're lucky, I guess. But having said that, we also try to repeat the tobacco studies or grafting. We did not see spread of silencing in tobacco.
I personally have a question, just to tie this together to the theme. If a student came to you with a similar, and I know you face some unique challenges with this project, but if someone came to you with a similar idea in this day and age, what would be kind of your advice at this point? your 1 minute answer to that.
Advice in terms of what? Whether to do something like this, or
what kind of impetus do you need to from the industry level to motivate a project that's going to end up being at some point very large and possibly business project?
Well, okay. If I did not see the implications, and it's huge implications, 500 million people, so protein source for 500 million people. If it didn't have that sort of impact or potential impact, we haven't impacted anything yet. If it didn't have that sort of potential impact, I probably would have, once we published the paper, we've moved on to something else. Having, as you probably can tell, I come from India, spent about 21 years in India, and I've seen poverty firsthand, and I've seen hungry people. I've seen people, who live on just bread alone, and so I've seen how people suffer from this protein malnutrition. Also, my father was a doctor, so he was seeing all these patients. A lot of the sickness comes from being hungry and not having enough nutrition. So maybe I'm a unique case in that respect that I didn't give up. Or many times I wanted to give up, but and uh, it's again, this is science, and you have to be a little bit of stubborn to be able to accomplish something. There is a very fine line between being totally foolish and being stubborn and all that. So uh, we, I was pretty hopeful that we'll be able to do it, and we could have done it sooner if we had funding right from the beginning. We just didn't have funding. In fact, there was about a period of 4 or 5 years we had no funding to do this work, and so during that time, that Dr. Borlaug also tried to help us, called some Rockefeller Foundation and so on to say, okay, can we get funding for this? And obviously we didn't get any. So that's why it took a little longer than it should have done.
Okay.
Thank you.
This one might not be working. This one's not. Is this one on?
That one's on.
It's not on, is it?
Okay, can you guys hear me? Okay, perfect. So next we'll be hearing from our fourth, excuse me,